Okay, welcome back. This is Issues in Biotechnology, the way we work with life. And we're into the medical biotechnology sector, looking at now emergent technologies in cancer treatment. We have already examined cancer basics, but now we want to turn our attention to the application of biotechnologies as they relate to treating the various diseases of cancer. Will there be a cure for cancer? Not one, but many. And owing to the fact that cancer is not one disease, but many, and caused by the many functions that we've already described. So let's look at the applications of biotechnology, but first give a brief history on cancer chemotherapy, as it is one of the triad of effective treatments, including also radiation therapy and surgery. But then I want to follow up with 3 case studies, actually 4, but 3 case studies specifically looking at DNA-based technologies, and then lastly, sort of forward-looking into the issues and opportunities for doing more in the future. We saw in the previous lecture the use of DNA microarrays and how this will play an important role in molecular staging and diagnostics, but using DNA technologies and specifically RNA technologies also give us targets for using RNA interference to tie up expression of genes specifically involved in cancer treatments. So we already described in a previous lecture how DNA gene chip technology works, shown again in this slide, comparing normal versus tumor cDNAs labeled with green for normal and red for tumor cells, and then putting them over a gene chip to see where tumor-specific RNAs are expressed. And here is a dramatic slide showing disease-free panel on the top versus a metastasizing tumor on the bottom relative to its gene expression levels. Pharmacogenetics, looking at the phenotype of certain cancer types is one thing, but looking at how people respond differently to different drugs is also very important for interrupting tumor cell growth. And looking at the analysis of RNA levels and protein levels will help us decide what drug might be good for you and do nothing for you. And certainly this will be relevant to certain types of molecular staging events. In other words, you both might have colon cancer, but you both might have a different type of molecular colon cancer. Responding to one type of drug might work for you and not for you. The same thing with pharmacogenomics. Understanding the genes which predispose certain people to certain cancers also might be relevant in deciding how to treat that disease or prevent it in the first place. Variation in phenotype is a combination of the variation in genetics by the variation in its environment. So learning more about cancer prevention is important also for how we treat this disease. We have already seen that smoking cessation has caused a dramatic decrease in lung and bronchial cancer. And the more we understand about environmental cues relative to genetic predisposition, we can prevent some cancers from occurring in the first place. So cancer phenotypes not only have a genetic predisposition, but also through field effect by environmental cues. What causes cancer? Well, certainly genetics and environment. But genetics mutations, oncogenes, aneuploidy, chromosomal rearrangements, etc. And that human cancer genes have been linked to oncogenesis. And this slide is a reiteration of what we saw in the previous lecture. So it's this stepwise genetic change that has to become the target of molecular biotechnology to treat the disease. Carcinogens, too many to name. By the Ames test, too many to eliminate. Which ones do you eliminate? I think we can be somewhat wise about this. You stop eating bread crust? I don't know, how extreme do you need to be? Stop eating food in general. But there are, I think, some prudent choices that can be made about lifestyle based on the epidemiology. We know that an increase in eating red meat, for example, has been linked to certain types of cancer. So you can review carcinogenicity against mutagenicity results from an Ames test and make prudent choices about your exposure to certain types of carcinogens. So what are the emergent technologies? First, a brief history on chemotherapy. The USS John Harvey was an American ship in the Bari Harbor, and it carried highly classified load of about 2,100-pound mustard gas bombs, and it was damaged during a German raid that damaged 17 ships, including the John Harvey, on December 2nd, 1943. A fire on the Harvey caused mustard gas-laden smoke to spread quickly, and about 617 gas poisoning cases among the troops and merchant marine seamen were reported, with a 14% fatality rate. That's threefold higher than that of World War I. And this incident is commonly cited as the key observation that led to the beginning of chemotherapy derived out of chemical components comprised in mustard gas. And you know how these conspiracy theories work. They usually implode from the bottom up, you know, exposed by their own weight. So that actually, when you look into the work by Goodman and Gilman, who were working at Yale's Department of Pharmacology, it was not until 1942, more than a year before the Bari raid, that these two were commissioned by the Defense Department. But it had been broadly reasoned that mustard gases could kill cancer cells in lymph nodes prior to that. So it was in December of 1942 that a 48-year-old patient with terminal lymphosarcoma was given 10 consecutive doses of nitrogen mustard components that resulted in the first demonstration of this as an effective chemotherapy. From the humble beginnings of a horrible mustard gas, the alkylating agents that have resulted in the structure of major nitrogen mustards and the result of different chemotherapies that have resulted in the slowing down of rapidly dividing cells. So the overall suppression of rapidly dividing cells is the outcome of treating with these types of drugs, owing to the side effects of all rapidly dividing cells in the body are then thus affected. So rapidly dividing cells in hair follicles and the side effect of hair loss subsequently, and we can imagine that other rapidly dividing cells within the body are subsequently affected by the systemic treatment of chemotherapy, although this has been an effective treatment demonstrated for many years. So the rate of clinically effective anti-tumor drugs is shown in this graph, rising from 1943 almost nearly exponentially to the current time. Cytotoxic drugs in 2012 are still among the standards of care in cancer therapy, even though we have now benefited from the development of recombinant DNA drugs and other procedures. Certainly these should not be neglected. In fact, for breast cancer, the use of Taxol, which is derived medicinally from a plant, a yew, a conifer, that has been shown to be very effective in treatment of breast cancer, coupled with Herceptin, a recombinant DNA drug. But the list of effective chemotherapies present in this list demonstrates, I think, the effectiveness of cytotoxic drugs that still remain one of the gold standards of treatment for many developing cancer types. The key limitation of cytotoxic chemotherapy depends on how fast that tumor is growing. Obviously, it's going to be more effective on cancers that are rapidly growing versus those that are more slowly growing. How fast is the tumor growing is one of the questions. Let me now turn to different case studies on new drugs developed out of biotechnology approaches that are making a difference. And this, I think, will shed some light on the future of new drug targeting in the post-genomic era. We have recognized that inhibition of angiogenesis is a specific target, that angiogenesis is required for tumor metastasis and growth beyond 1 millimeter volumes, as demonstrated in many different types of cancer, including breast cancer, and that there is a broad spectrum of activity that results in angiogenesis. Folkman was the first to describe as angiogenesis as being necessary for tumor metastasis, and that access to vital supplies, nutrient as well as growth factors, are supplied by blood vessels stimulated towards growing tumors. Tumor growth is limited by access to nutrients and waste removal mechanisms. And the tumors can stimulate blood vessel growth specifically by hormones which trigger angiogenesis. So blood vessels grow towards a tumor based on hormones or growth factors produced by the tumor itself. Remember I said the tumors are smart? They're smart at the molecular level, creating growth factor hormones to attract blood vessels that further the growth of those own cells. So vascular epithelial growth factor, VEGF, is one of those growth factors which stimulates angiogenesis. So blood vessels shown in this photograph grow towards a sarcoma tumor shown in this rat muscle tissue. Before I get to recombinant DNA drugs, protein-based drugs, first we have to recognize that DNA technology has also been important to the development of small molecule drug design. I already discussed the role of small molecule drug design in the pharmacogenomics section of this course, but certainly it's also relevant to being able to design certain molecules that will uniquely fit into receptors such as VEGF or other types of receptors or proteins involved in signal transduction pathways in cancer. Small molecule drug design will continue to be important, even though I am focusing in this class on DNA-based drugs. Certainly the advent of Gleevec and its FDA approval in 2001 has demonstrated the effectivity of small molecule drug design for controlling certain types of cancer. So recombinant DNA drugs and will create new cancer drugs, not only based on its targets, but based on the biology of those signal transduction pathways. So we can expect that new precise treatments for cancers will be developed on protein-based drugs, not relying anymore on systemic treatments that simply slow down growth, but actually protein-based drugs which will attach specifically to their targets. This should eliminate, or at least greatly reduce, side effects that are incurred by normal chemotherapies. Rather than shutting down cell pathways globally in the system, limiting or targeting drugs. Moreover, attaching drugs, small molecules, to antibodies that recognize cancer cells only should further limit systemic chemotherapeutic side effects. So you've seen this diagram before. Our ability to construct transgenes, artificial genes comprising promoters that control expression for making certain proteins, downstream coding sequences that code for specific proteins, and termination signals allow us the ability to create new proteins de novo in cell systems outside the human. So in the example of Enbrel, for example, we made a protein as an antibody that would recognize proteins that are involved in the autoimmune disease for tumor necrosis factor alpha, stimulating then rheumatoid arthritis. Similarly, we can make antibodies that will recognize proteins and receptors involved in carcinogenesis. And here's just a list of 4: Erbitux, Herceptin, Avastin, and Rituxan that have been effective antibody-based drugs that have been made in this way. These then would be injectable drugs that would have a longer half-life than small molecule drugs, typically. Here's an example of Avastin, how it could block angiogenesis by having the antibodies react specifically with VEGF, and therefore not allowing VEGF to bind to cancer cells, stimulating angiogenesis. So by blocking VEGF secretion, you suppress angiogenesis and therefore limit tumor growth. And this has been a very effective drug for treating certain types of cancers. So why develop an antibody? Well, VEGF was demonstrated since 1996 to be involved in angiogenesis, and therefore became a specific target by developing antibodies that resulted in the creation of Avastin. This is intelligent drug design specifically against targets. Previously, a lot of small molecule chemotherapies were developed out of a spray and pray approach, looking at drugs that slow down cell division, like mustard gas. Well, yeah, you can slow down cell division, but look at the other horrible side effects that are involved. The good news is you survive cancer. The bad news is you survive cancer. You know, that the side effects of chemotherapy by some patients has been viewed as one of the most difficult aspects of the disease itself. The advantages for antibodies are that they're ideal for extracellular targets. They're not going to see intracellular targets, but if you've got something sticking out through the membrane, these proteins become very effective targets. And whereas small molecule approaches are successful for beautiful binding sites, specifically on outside receptors, but are generally limited for all others, including targets that you might want to recognize like non-enzymes or non-receptors or shallow pocket enzymes that small molecules may not be able to see. It also allows for this exquisite selectivity that an antibody is going to see maybe even only one epitope of a receptor that is sticking out of that membrane. Another advantage is that they have relatively long duration of coverage over a target tissue, that its half-life for a human antibody may be weeks versus hours for other small drugs, which many chemotherapeutic patients have to continuously take small molecule drugs, also increasing the likelihood of side effects and cost. The potential risk or the distribution of antibodies may limit the activity relative to small molecules. And there is the potential for immunogenicity. You could become allergic to the very antibody that you are delivering. This has occurred, going back to my Enbrel example, in patients that are treated for psoriasis can develop an allergy against Enbrel itself, rendering that drug less and less effective. Manufacturing can be more complicated. It will involve that same sort of biomanufacturing process and therefore increasing the price of such treatments. However, if you're dying of cancer, if you have the proper health care, and that's another debatable system right now, certainly the Supreme Court weighed in on that, I think, today. We just saw a largely Republican Supreme Court maintain the Obamacare health law. Another disadvantage is that human antibodies rarely cross-react with rodents, so preclinical modeling becomes challenging. We need a large number of patients. I can also tell you a story based on a company called Large Scale Biologics that was in California and developing antibodies in plant systems, so-called plantibodies. So here's a system that's outside of an animal cell-based system. Humans do not get pathogens derived from plants, so the system itself very good. Their idea and their business model was to take tumors from a patient, develop antibodies in a rabbit or a mouse model, develop those antibodies very quickly, sequence them, get the DNA specific for that antibody. So now we have a sequence for DNA specific for an antibody against your tumor, not your tumor, your tumor. Now cloning that fragment of DNA into tobacco mosaic virus. Now we're going to use tobacco to cure cancer by infecting a tobacco field with this genetically modified virus. The virus infects the tobacco, makes a huge amount of this antibody specific against your tumor. This all occurs in weeks. So we take your tumor, biopsy it, make the antibody. That takes 2 to 6 weeks. Clone the fragment of DNA, 9 days. Take that, put it into a TMV vector. Spray a field of tobacco, harvest the protein another 4 days. 2 months go by, I have an antibody specific to your tumor. Great. It worked. They actually treated people and this worked. And then the company went bankrupt. Because you can't get FDA approval on a personalized antibody. What a bummer, right? How do you do clinical trials on a single antibody against a single person? We need to question, I think, this whole idea because it will jeopardize personalized medicine in this way and limit some of these therapies. Nonetheless, there are large classes of therapeutic antibodies that have been developed for cancer, and here's a list of those products that I alluded to earlier, what their target proteins are in the companies that's developed them. You may remember Erbitux, developed by ImClone, as the famous protein that sent Martha Stewart to the slammer. She got wind of a failed phase 3 clinical trial, sold her stocks based on insider knowledge, and that insider knowledge sent her to jail. Meanwhile, the phase 3 clinical trial was to show that it was going to fail, so she sold her stock before the stock went down with that news, and therefore that was viewed as insider trading. In a subsequent trial of Erbitux, it actually worked, and now it is on the market, and Martha Stewart's out of jail. Not because it worked, but you know, it's interesting how things play out. The advantages to Avastin is that it's one of the first line approaches in many different types of breast cancer now, and has also been used to treat lung cancer for its preventative applications against angiogenesis. Herceptin actually has a humble story beginning in carcinogenesis discoveries in chickens, but Herceptin binds to the outside of a receptor called IVb.2, which is present in some types of breast cancer. HER2 types of diagnostics have been used to identify epithelial growth receptors such as EGFR, which is a family of transmembrane kinases involved in certain types of breast cancer. If you're identified as a patient with breast cancer in which HER2 is amplified, then you are a very good candidate for Herceptin. And here is a checklist of why ERBB2 is a good target for Herceptin as an antibody to interrupt that function. Herceptin is a monoclonal antibody, and I described in an earlier lecture how monoclonal antibodies are actually made. But by recognizing the outside of this receptor and blocking its function, we block the progression of that type of breast cancer very effectively. So this is really a poster child, if you will, for the development of these types of monoclonal antibodies that will recognize extracellular receptors involved in cancer, limiting the systemic side effects that would be present in treating these. So with the use of still surgery, small molecule drugs like Taxol, together with this treatment, cures against HER2 type breast cancers are extraordinarily high. And this table shows the clinical activity of breast cancer related to treatments with Herceptin. And the activity has improved survival as demonstrated in this data in these graphs here. So on the basis of these results, the ability to care for patients with HER2 positive breast cancer has changed dramatically. And certainly patients with lymph node positive HER2 breast cancer should receive these types of drugs as an optimal part of systemic other therapies. So many of the drugs used in chemotherapy now are, and you can decide what the answers to that might be. So let's look at other therapeutic antibodies that are used for cancer. Antibodies have this distinct advantage over old school chemotherapies for fighting cancer because they, and again, you can evaluate those based on what we've previously gone over. Another interesting drug in our case studies here today it involves Tarceva, which, unlike Herceptin, which acts on exterior receptor sites, Tarceva is targeting the ERBB1 factor that's internal. So overexpression of this target, EGFR, for anti-tumor drug discovery is based on the ability to limit overexpression of that target, which transforms cells in a ligand-independent manner. So knockout mice of these were available that lacked the EGFR target and were used early on to validate this as an anti-tumor drug target site. And I think this underscores the use of mouse as a human cancer model. We have already underscored the importance of mouse as a genetic model for humans, and certainly using it as a cancer model in the case of Tarceva proved to be invaluable. Certainly the use of mouse genomics as a human cancer model is also important, and the ability to use that genetics to study human cancer targets is proving to be hugely beneficial. The so-called onco mouse was developed at Harvard as a mouse target that has the capability of developing many different types of cancer for which different treatments can be evolved. So the ability to do cancer treatments on the onco mouse relative to treating drugs prior to human treatment, as shown here in the treatment of a female mouse looking at progression under CT scans, has been very valuable for the development of new drug targets. Certainly the pharmacological selectivity of close oncogene kindreds will also be important for the development of new site-specific small molecule drugs, as shown here in the green part of this diagram on the upper left. Tarceva then has advantages in that it has intracellular targets, namely the tyrosine kinase active sites, with exquisite selectivity. And it has a relatively long duration of coverage in comparison to what we might see for other types of treatments. So new frontiers, certainly the list is as long as the cancer gene list that I showed in lecture 1. What we see now, to summarize the second part of this lecture, is that targeted therapies, those abrogating the key molecular advantages of tumor cells, are adding importantly to patient survival. And I think we can continue to see progress in this area. These approaches offer promise of less morbidity and higher quality of life as people maintain life with cancer. Certainly being diagnosed with cancer probably then becomes a life-changing event no matter what. Monoclonal antibodies have been credentialed targeted therapies, and I think through this development we will see more antibody-based drugs, hopefully rekindling personalized antibody development, such as the example that I alluded to earlier. I think also personalized RNAi approaches, nanotechnologies that will couple antibodies and drugs to either small magnetic particles or particles that are directed specifically to cancer cells will further limit the nonspecific chemotherapeutic side effects that we've seen in the past. So the future, I think, is bright. Have we cured cancer? No. But our knowledge base, I think, will increase survival rate and certainly early diagnosis into the future. Thanks. The clear bead at the center changes everything. There are no edges to my being now. I have heard it said that there is a window that opens from one mind into another. But where there are no walls, there is no need for a window or fitting a latch. That's a poem by Rumi, 1279 or so. Thanks for your time and attention.